In a 2011 study at the University of Rochester Medical Center, researchers measured a plant-derived compound called parthenolide against ibuprofen in inhibiting NF-kappa B, the master inflammation switch inside human cells. Parthenolide blocked the inflammatory cascade at concentrations roughly 1,000 times lower than ibuprofen required. The plant grows wild along roadsides and abandoned fields across North America and Europe. It is called feverfew, Tanacetum parthenium. Yet no major pharmaceutical company has brought a parthenolide drug to market in the 50 years since its mechanism was first described. The reason is not safety. The reason is structural. The molecule cannot be patented in its native form. Pedanius Dioscorides, the Greek physician and pharmacologist, compiled his Great Materia Medica around 50 CE. In Book 5, he lists a field herb called parthenion and writes that it is used for the diminution of fevers, for the relief of pains of the head, and for the curbing of the hardness of the mind. This is the earliest surviving record of feverfew's medicinal use, anchoring its reputation for treating headaches and fevers at the very start of the Western medical tradition. Centuries later, the Benedictine monks of St. Gall, writing in the early 9th century, catalogued feverfew among the 18 essential herbs of their infirmary gardens. Their Regulae Medicae, dated to around 820 CE, notes that they keep parthenium for maladies of the head and for fevers. The plant's name, and its association with head pain, carried through the margins of medieval manuscripts. A scribe at St. Gall even recorded the legend that feverfew was named after a workman who survived a fall from the Parthenon, and that the herb was then planted for the protection of the head. Between Dioscorides' clinical precision and the monastic scribe's practical lists, a continuous thread connects feverfew's ancient field use to its place in the structured medicine of early Europe. These records do not treat feverfew as folklore or rumor. Instead, they preserve it as a core remedy, documented and handed down by those charged with caring for the sick. The evidence is clear. Long before the age of patents and pharmaceutical brands, feverfew's value was already written into the fabric of Western medicine. By the late Renaissance, feverfew had moved from monastic gardens into the printed pages of European herbals. In 1597, John Gerhardt, the London apothecary and botanist, described feverfew as very good for them that are giddy in the head, capturing its reputation as a household remedy for headaches and dizziness. Gerhardt's Herbal, a thick folio crowded with woodcuts and plain-spoken advice, reflected the plant's place on the kitchen shelf as much as in the apothecary's jar. Feverfew was not rare or exotic. It grew along hedgerows and in cottage plots, its white flowers as familiar as chamomile or tansy. The tradition crossed the Atlantic. By the mid-19th century, feverfew appeared in the 1849 King's American Dispensatory, listed for fever, arthritis, menstrual pain, and chronic headache. The dispensatory offered precise instructions, dried herb prepared as an infusion or tincture, for intermittent fevers and migraine-type pains. Its inclusion in such references signaled that feverfew was considered reliable, effective, and safe enough for routine use long before laboratory isolation of its active compounds. In this print era, the authority of feverfew came not from patents or proprietary blends, but from centuries of recorded experience. Recipes passed from parent to child, reinforced by the printed word, kept the plant in daily medical practice. This domestic familiarity would soon face new pressures as industrial chemistry and regulatory standards began to redraw the boundaries of what counted as medicine. In 1899, Bayer secured a patent for acetylsalicylic acid, better known as aspirin. Their application described a synthetic process that transformed salicylic acid, a compound long extracted from willow bark, into a stable, crystalline drug. This was more than a chemical breakthrough. By patenting the new molecule and branding it as aspirin, Bayer claimed exclusive rights to its manufacture and sale. The company's marketing engine turned aspirin into a household name, overshadowing the field plants and folk remedies that had filled medicine chests for centuries. The shift was reinforced by the Pure Food and Drug Act of 1906. For the first time, federal law required medicines to be labeled accurately, with ingredient lists and purity standards. Small producers of herbal remedies, including feverfew, faced costs they could not absorb, such as chemical assays, standardized packaging, and compliance with new regulations. Many simply disappeared from the market. By 1925, feverfew was gone from the United States pharmacopoeia. Official records cite insufficient monographic data and declining commercial availability, but the reality was economic. Without the resources to meet regulatory demands or the protection of a patent, feverfew and other botanicals retreated from mainstream medicine. In their place stood branded synthetics promoted by companies with the means to navigate and shape the new regulatory landscape. In 1960, chemists at the University of Bonn succeeded in isolating parthenolide, the active sesquiterpene lactone, from feverfew. Their work finally revealed the molecule responsible for centuries of reported effects against fever and headache. Yet this scientific achievement arrived into a world where the rules of medicine had changed. Patent law in both the United States and Europe now drew a hard line. A molecule found in nature, even if newly isolated and characterized, could not be claimed as intellectual property. The so-called product of nature doctrine meant that parthenolide, unlike acetyl salicylic acid or ibuprofen, was ineligible for the legal protections that drive pharmaceutical investment. Drug companies faced a dilemma. Without the possibility of exclusivity, there was no realistic path to recoup the enormous costs of regulatory approval. The search for a workaround led to chemical modification. One such attempt was DMAPT, dimethylaminoparthenolide, a synthetic derivative patented in the early 2000s. DMAPT showed promising activity in preclinical cancer models and, crucially, could be patented because it did not occur in nature. But when DMAPT reached human trials, it faltered. The compound's rapid metabolism resulted in poor plasma levels, and the clinical benefits seen in the lab could not be reproduced in patients. The economic logic that had erased feverfew from the pharmacopoeia now blocked its active principle as well. The native molecule was too common to own, its analog too flawed to sell. In the end, the legal and financial structure of modern drug development left parthenolide and feverfew adrift, scientifically intriguing but commercially invisible. A double-blind randomized controlled trial published in 2005 put feverfew's traditional reputation to the test. Researchers enrolled 170 adults diagnosed with episodic migraine and assigned them to receive either a standardized feverfew extract, carefully measured to deliver a consistent parthenolide dose each day, or an identical placebo. Over 16 weeks, participants tracked their migraine frequency and severity in detailed headache diaries. The results were not subtle. Those taking the feverfew extract experienced an average reduction of 1.9 migraine days per month compared to baseline, while the placebo group saw a much smaller change. The difference was statistically significant. Side effects were mild, with about 1 in 10 reporting minor mouth irritation. No serious adverse events were recorded. This trial translated centuries of anecdote and herbal tradition into modern clinical data, showing that a rigorously prepared feverfew extract could offer measurable relief to migraine sufferers, without recourse to synthetic drugs or proprietary blends. A 2018 review published in Inflammopharmacology compiled more than 40 peer-reviewed studies on parthenolide, documenting its effects across inflammation, cancer biology, and neuroprotection. The authors described parthenolide's ability to block NF-kappa B, called the master switch of inflammation, at concentrations far lower than many conventional drugs. This broad mechanistic profile has drawn researchers from multiple fields, but the review also sounded a note of caution. About 1 in 10 participants in clinical trials developed mild mouth ulcers, especially when chewing raw feverfew leaves. Reports of gastrointestinal upset and rare allergic reactions have also surfaced. The review highlighted important safety boundaries. Feverfew should not be taken during pregnancy due to possible uterine effects, and it may increase bleeding risk for those on anticoagulant medications. The evidence does not support using feverfew as a replacement for emergency care or prescribed treatment without a physician's oversight. The scientific record as synthesized by the review is both promising and clear-eyed. Parthenolide offers multi-domain effects, but its use requires the same vigilance as any active medicine. In 2011, a team at the University of Rochester led by Dr. Craig Jordan put parthenolide's reputation to the test in a direct comparison with ibuprofen. Their laboratory work focused on the NF-kappa B pathway, a central switch in the body's inflammatory response. Parthenolide, a sesquiterpene lactone found in feverfew, blocked NF-kappa B activation in cultured cells at concentrations measured in nanomoles, about 1,000 times lower than the amount of ibuprofen needed for the same effect. The experiment used standard luciferase reporter assays to track NF-kappa B, confirming that parthenolide's inhibition was both potent and dose-dependent. This finding gave scientific weight to centuries of folk use, but it also exposed a legal obstacle. Because parthenolide is a natural molecule, patent law prevents any company from claiming exclusive rights to it. The same chemical structure that grants its biological power also ensures it remains beyond the reach of pharmaceutical ownership. For 2 millennia, from Dioscorides' pains of the head to Gerhardt's giddy in the head, feverfew's value was common knowledge. Today, its absence is not the result of scientific consensus, but an artifact of patent law and regulatory cost. The remedy remains, if the system allows us to remember. Welcome to Loamhold. Subscribe for more.